Today, I have the pleasure of introducing Daniel Batanau. Today, he's going to give his final master's seminar titled CHROMOMETHYLASES in the DNA methylation dynamics of cold responses in Brachypodium distachyon. Daniel graduated from McMaster University in 2021 with a bachelor's in biology, focusing on molecular biology and genetics. And while at McMaster, Daniel completed his thesis studying the expression of late embryogenesis abundance proteins in response to recurrent drought stress. In 2021, Daniel joined Dr. Jean-Benoit Charon's lab to investigate the influence of epigenetics on abiotic stress in Brachypodium distachyon. Daniel's research interests lie in how genome editing can be used to develop stress-tolerant crops and how the different levels of biology communicate with one another. With that, to help the floor.
Thank you. Good morning, everyone. Can everyone hear me? Can I just get like a thumbs up or an audio confirmation from? Awesome. Thank you, Luke. Okay. Good morning, everyone. Thank you for coming, especially in these weather conditions, those both in person and online. I'll be presenting my my final thesis. So let's begin. And there we go. I'm very sorry. More.
Yeah, at the far end.
Yeah.
And then go to a high protein. Well, yeah.
Got it. Okay. Sorry about that, everyone. Okay, so to begin, late spring and early fall frost events are some of the most damaging abiotic stress events in agriculture. Reason being, the timing of these stresses coincide with early and late developmental stages of crops, and plants that germinate at this time are typically important agricultural products. An example being spring wheat, which accounts for about 20% of Canada's agricultural land use area. Concern over the security of staple crops has been raised as the frequency and severity of frost events is expected to increase as climate change continues. I don't know if this is considered a late fall frost event, but we did have some pretty funky weather today. Okay. Nope. So cold stress occurs when environmental temperatures deviate below their natural range, and the severity, frequency, and duration of the stresses determine its impact on the plant. Cold stress typically leads to a reduction in enzyme kinematics, slowing down and reducing the cellular machinery. We additionally see an alteration in flowering, a reduced leaf expansion, and an overall reduction in growth. At freezing temperatures, we begin to see cellular damage and dehydration, which further exacerbates the stress that the plant is experiencing. So plants are sessile organisms and they can't move, but plants are clever and they've adapted a series of mechanisms in order to adapt to their environmental temperatures. In response to cold, plants will trigger a signal cascade, which leads to the adaptation to reduce temperatures. This begins first through the detection of the deviation of environmental temperatures, leading to an activation of signal transducers, such as cold-1. These proteins activate transcriptional factors, such as the CBF family and ICE1, which induce the transcription of cold responsive genes, leading to a cold stress response. Phenotypic changes in response to cold can be quantified within 1 hour of cold exposure, which is quite rapid. Short-term cold exposure limits growth and development in exchange for survival in these conditions. So there's a bit of a trade-off. Prolonged exposure to cold, though, increases a plant's capability to survive in reduced temperatures and even grow through a process which is termed cold acclimation, in which a plant adjusts its phenotype and primes itself to better tolerate recurring cold stresses. So the transcription of these cold responsive genes and general transcription in plants themselves is very tightly orchestrated in order to create a precise response in the environment. One of the largest studied fields of transcriptional control is epigenetics, and epigenetics may be defined as changes in the phenotype or expression of a gene or phenotype without alteration of the genomic code itself. These changes are heritable and reversible and typically generate a rapid and sustained response. Transcription control through epigenetics is present in all levels of DNA, including at the chromosome level, the chromatin level, the histone level, in which marks are deposited on histone tails. And today, what we're going to be focusing on, which is epigenetics. Now, DNA methylation. Now, this ability to fine-tune its expression increases the range of phenotypic conformations that a plant can take, which we could consider phenotypic plasticity. So this level of regulation of epigenetics kind of controls and increases that range of conformations that an organism can take in response to not only stresses, but stimuli. Now, as I said, today we're going to be focusing on DNA methylation, and this is one of the methods in which epigenetics regulates transcriptional control. DNA methylation itself is a form of epigenetic regulation in which methyl groups are directly deposited onto DNA to control the transcription of genes. As we can see here, we kind of have a cytosine, unmethylated cytosine, and then right here we have a methylated cytosine where on the C5 carbon we have an additional methyl group. Now, it's deposited in DNA, and this is commonly associated with transcriptional silencing, but actually the conformation and the location of these methyl marks actually creates kind of different expression patterns. So on the right here, commonly what we see is if we see DNA methylation in the promoter region, these little marks right here are methylated cytosines, by the way. We typically see transcriptional repression. Meanwhile, when we see methylation marks on the coding sequence or body of the gene, where the exons and introns are, we typically see constitutive expression and kind of a continuous controlled expression. This is called gene body methylation, and you can typically find it on genes that are important to the organism that are constitutively expressed. And finally, one of our examples is transposon silencing, where you'll see a methylation all across the transposon itself, and that is meant to silence and prevent the expression of the gene and prevent transposons. What they like to do is kind of jump, so it prevents that. Now, methylation is tissue dependent. You will see different methylation profiles in different tissues of the organism, and that kind of relates to that fine tuning of the different organisms. And this is especially important. Methylation itself is important for complex genomes, particularly those that are polyploidy and have multiple copies of the same gene, in order to ensure that there isn't random and kind of uncontrolled expression. So today we're primarily going to be focusing on DNA maintenance, but the actors involved in DNA methylation can be categorized into three groups: readers, writers, and erasers. Readers being those that can detect methylation marks, writers that are capable of depositing methylation marks, and erasers that are able to remove methylation marks. So methylation is commonly deposited through the RNA-directed DNA methylation pathway, and excision in plants is done by the Demeter family, while methylation maintenance is primarily done by genes that have chromodomain proteins. So in this figure here, we kind of see the de novo methylation itself, and then here we see those actors involved in maintenance. And what you see is there's specific patterns associated, so CG, CHG, and CHH pattern methylation. And what we see is that there are specific proteins that are involved in those different methylation patterns. So in CG, we commonly see MET1, CHG, we see CMT3 and CMT2, and CHH, we see DRM2 and CMT2. Now, right here, I would like you to focus on, there is actually an interplay between both histone modification and DNA methylation, in which the SUVH family proteins, which are histone modifiers, actually require CHG-style DNA methylation to bind and create H3K9 dimethylation. Meanwhile, CMT3 and CMT2 require H3K9 dimethylation to bind and create CHG methylation on DNA. So there is an interplay between both histone modification and DNA methylation. What I primarily focus on is a group of actors involved in DNA methylation known as the CHROMOMETHYLASE family. These proteins assist in the deposition and maintenance of DNA methylation marks and are characterized by the presence of a chromodomain, which is DNA methyltransferase, and a BAH domain, which is involved in protein-protein interactions, recognition of methylated histones, and nucleosome binding. So, yeah. All right. So previous work has established that there is evidence that genetic cold acclimation is partially regulated by epigenetic modifiers, but there is a knowledge gap between the impact of DNA methylation actors and cold acclimation. Now, this leads me to the aim of my study, which is to investigate the role of the CMT family in the monographs Brachypodium distachyon in response to cold stress. The reason why we study Brachypodium distachyon, it's recently, over the past decade, it's kind of been put at the forefront as a model organism for grasses. Reason being is its small stature. It is a diploid organism. Meanwhile, many grasses are polyploidy, which makes it easier to study. It has a small genome and a rapid life cycle of about 4 weeks. And this leads me to my hypothesis that disrupting DNA methylation through mutation of CHROMOMETHYLASES genes will lead to alterations in the cold stress response of my model organism, Brachypodium distachyon. So my project consists mainly of 4 primary objectives. First being the characterization of CMT genes, of Brachypodium distachyon CHROMOMETHYLASE genes. Analyzing the transcriptomic accumulation profile of Brachypodium distachyon CMT genes in response to both short and long-term cold stresses. A sequence validation of Brachypodium distachyon CHROMOMETHYLASE genes that contain mutations in those CMT lines, and a phenotypic characterization of said mutant lines. So in my first objective, as I said, the purpose is to survey the Brachypodium distachyon genome for CHROMOMETHYLASE genes. The way I did this is I did a protein domain search through Interpro for genes that contain a chromodomain in Brachypodium distachyon. I then refined the search, looking for proteins that have the presence of both a BH domain and a C5 DNA methyltransferase domain. Now, this led me to refine the results to about 4 CHROMOMETHYLASE genes. Now, this objective was to verify the this objective, or in this purpose of this, was then to verify the organization of the domains in each of the Brachypodium distachyon genes. Through this, I used an NCBI conserved domain search in order to predict possible domains within each of those 4 CHROMOMETHYLASE genes. Now, what we see here is we see a relatively common organization. Now, this is the predicted protein structure of the Brachypodium distachyon genome. So here we have our amino acid length. We have CMT1, CMT2, CMT3A and B, which are paralogs of each other. And as we can see, the organization of this is quite similar among all those 4 CHROMOMETHYLASE genes. We have our bromo-adjacent homology domain, which, as I said, is involved in protein-protein interactions, recognition of methylated histones and nucleosome binding. Our chromo domain, which also binds to histones and nucleosomes and mediates protein-protein interactions, as well as our C5-specific DNA methylase, which is actually what does that deposition of that methyl mark itself. So we also kind of see, interestingly here, an STK predicted to have an cytosine threonine kinase catalytic domain. It's only predicted, and it's only the catalytic domain, and this suggests that it might be an evolutionary relic or a off prediction. But moving on. So in conclusion, this kind of verifies that we have 4 Brachypodium distachyon CMT genes that fit that classification. Now, this next step was to investigate the relationship between CMT proteins in Brachypodium distachyon and other monocots. The methods I did this, I did a protein blast using my Brachypodium distachyon CMT genes as a reference. And I used species such as Brachypodium distachyon, Brachypodium stacei, Oryza sativa or rice, Z. maize corn, Ornopintium thoramium, Panicum hali, Panicum varangium, which is switchgrass, Stella italica, which is millet, foxtail millet, and sorghum. Now, I found those genes. I once again refined my search in order to verify that we have those BH domains, that chromo domain, and C5 catalytic domain. And then I constructed a sequence alignment and did a phylogenetic construction using Mega 11, using the maximum likelihood method with 1,000 bootstraps. Now, what we see here is it quite nicely clusters. We have our CMT1 genes that grouped, our CMT2 genes that grouped right here, and our CMT3 genes that grouped right there. Interestingly, we have Z. maize and CMT1 kind of clustered together here on their own graph, which is quite interesting. But what we have is we have nice bootstraps and kind of clustered together, and this kind of verifies that the Brachypodium distachyon CMT genes that we have both cluster within monocots and kind of reinforce the ability that these are in fact CMT genes. Our next step, the next thing we move on to objective 2, which is a characterization of the transcriptomic accumulations in response to cold stress. And what we want to do is we first wanted to evaluate the response of Brachypodium distachyon genes through a cold expression analysis prior to that. So luckily, over the summer, the Joint Genome Institute released something called the Gene Atlas, which has a massive transcriptomic profile of multiple organisms in response to stress. So this massive RNA-seq library. So we took that and we found the Brachypodium distachyon CMT genes of interest, and we graphed them using that RNA-seq data through those FPMK values. So what we see here is an exposure of cold stress over a period of 49 days. And we have our CMT genes of interest right here. So what we see here, CMT1 appears that it's quite not there. The expression of the CMT1 gene is relatively low to the point where it appears like it's nothing there. In CMT2, we have a general, in the first day, we have relatively consistent expression, but around 14 days, we begin to see the expression or an increase in FPMK values of the CMT2 gene over a course of 14 days, which is kind of aligned with the vernalization, beginning of the vernalization response as well. CMT3A is relatively, there isn't any direct trend. Meanwhile, CMT3B, we kind of see a general downward trend in FPMK values over the course of the 49 days. So this is quite promising, and the virtual expression suggests there is some sort of alteration or response to cold stress through our CMT genes. Moving on, we wanted to determine the impacts of short and long-term cold stress in the transcription of Brachypodium distachyon CMT genes. So the way that we did this is we did a RT-qPCR analysis using relative quantification in the delta-delta CT method. The way I did this is I exposed plants to 24 hours of cold exposure at 4 degrees Celsius. I harvested the samples at specific time points. I extracted RNA, converted it to cDNA. Then I designed primers for both the CMT genes as well as common cold responsive genes and optimized those. And then I conducted qPCR and generated these graphs using the delta-delta CT method. So our first graph right here that I would like to put your focus on is COR410, which is a gene that is a dehydrin that is known to be responsive to cold stress. And we see quite a substantial increase in cold expression, about a 70-fold increase relative to control conditions. Also, the control conditions are plants are kept at 22 degrees over the same period of 24 hours, and we sample it at the same time. So we do have a cold response, and that verifies that. Our CMT3A, CMT3B, and CMT3-2 genes don't show too much of a substantial change in expression over that 24-hour period, which is quite disappointing. And even then, we do see quite a variable expression rate. But this is kind of expected because we don't expect to see substantial changes in methylation profiles in response to such short-term cold stress. We are currently doing a long-term cold stress experiment. However, due to the current renovations and closure of the Brachypodium, I've had quite a bit of difficulty doing this experiment. Fortunately enough, I have obtained a 4-degree growth chamber that can sustain that temperature for over the course of a month. And I am almost done sampling. So next week I'll be done sampling and I can move on to the qPCR experiments. So kind of what we can conclude from here is that Brachypodium distachyon CMT genes are not transcriptionally responsive to short-term cold exposure. So this in our next objective 3 is we wanted to observe the impacts of a mutated copy of CMT genes in Brachypodium distachyon. So the Joint Genome Institute has a chemical mutagenesis library of SNPs. And we looked at this tilling library, it's called a tilling library, which has that range of SNPs. And we identified 16 lines of interest to us. Now, this tilling line has a series of point mutations scattered throughout the entirety of the genome. So you select kind of SNPs that you think you're interested in. And those SNPs can either generate a silent mutation, nonsense mutation, or a missense mutation, or a splice site, splice site damage, or mutation of splice site, which would hypothetically create a non-functional or knockdown line or a less effective CMT gene. So we found 16 lines that interest us that contain mutations in one of those 3 domains of CMT genes that would either result in a missense, nonsense, or splice site mutation. And our strategy here was to get a series of about 5 lines per gene and kind of create a guilt by association technique where if we see kind of similar phenotypic pattern between all of our 5 lines, we can assume that that is a consistent pattern that can be associated with the mutation of the CMT, mutation in the CMT gene. So if it talks like a duck and it walks like a duck, it's a duck kind of situation. So what we see here is kind of our list of those lines that we have. And as you can see, we have targeted domains in both the methylase group BH domain and the chromo domain. So the way I did this is I grew the plants, I extracted genomic DNA, I PCR'd a region which highlights the SNP. I put this into a vector, I transformed it chemically into E. coli, I purified the plasma and I sent it out for sequencing. And this is kind of what was returned. So I got this alignment. I aligned the the CMT gene of interest in my mutant line, and I got one little SNP in the entirety of the genome. And this is what I was looking for. So the sequencing was completed by Genome Quebec, and these sequences were analyzed by Mutalign. Unfortunately, only 9 out of the 15 sequences actually contain the SNP of interest. But fortunately enough, we have a tDNA Brachypodium distachyon CMT line. So we have a knockdown line that has from a previous project that has a tDNA line inserted into the CMT3B protein or the CMT3 gene, and we will be using that as well for our project. And in the interest of this project, we're only going to be, in the interest of time, we're only focusing on the CMT3 line, so CMT3B and CMT3A. And now finally, this final objective is the phenotypic analysis of these sodium azide lines in Brachypodium distachyon. We want to characterize the role of each CMT gene in response to cold stress in Brachypodium distachyon. And we're going to be doing similar experiments what we did in our previous objectives, such as an RT-qPCR quantification of long-term cold exposure. And we're going to be using multiple cold responsive genes that are going to be used as markers. So that includes Core410, VRN1, CBF genes in order to ensure that there is a response and see if there's any deviations in that response between our wild type lines and our mutant lines. In addition, we're going to be doing a phenotypic characterization. We'll be taking about 10 plants from each line. And I'll be measuring the plant height, seed number, number of tellers, and seed weight, as well as conducting a fluorochem experiment to monitor fluorescence, chlorophyll fluorescence kinetics in between the wild type and our non-mutant lines. So in conclusion, we kind of found that Brachypodium distachyon has 4 chromomethylase family genes. Brachypodium does not increase expression of CMT gene in response to short-term stress, but we are currently looking at long-term stress. The Brachypodium distachyon sodium azide mutant lines contain mutations in key domains of Brachypodium distachyon genes. Once again, our current ongoing experiments are the phenotyping experiments on the long-term cold exposure and some future directions that can be taken. Do the methylation profiles around these cold responsive genes that we're looking at change during abiotic stress, as well as a whole plant freeze test in order to gauge the survivability of these mutant lines to our relative to wild type plants. So I would just like to acknowledge the members of the Sharon lab, including my PI, Dr. Jean-Benoit Sharon, Luc Olette, Basim Das, and our former postdoc, Kuna Kapoor, my committee member, Valerio Hoyos Villegas, the Geitman lab, who we share a lab with, who kind of made my days a little bit brighter, the Joint Genome Institute for providing seeds, Santra Sev, and the genome editing for food security and environmental sustainability cohort for providing funding. Thank you so much for listening. Do you have any questions?
Okay, so we've got online questions. If there are any questions here, try to moderate. Devon.
Nice presentation.
Thank you.
Just curious, thinking about long-term exposure,
Mhm.
do you think these responses will be different across different development stages?
Can you repeat the question for people?
Yeah. So the question was, will there be changes in methylation profiles and development in different tissue types and developmental stages? Yes, definitely. I'm personally only looking at leaf tissue, but you do see differences in methylation profiles across different tissue types, even in Brachypodium distachyon. You can see a lot of online resources. But in particular, CMT1, which is in my project, we kind of see fairly low expression all across the throughout the experiments. However, it's especially active in seeds. So you do see massive changes in methylation profiles in seeds. So that would be somewhere along those. Changes in that methylation profile maintenance are typically associated with seeds, but we're looking at leaf and we're looking at leaves and the actual life cycle of the plant. So in the tissue. Yeah. Thank you for the question. Anyone? Yeah.
So you've shown your RT-PCR graph and you didn't get like a lot of change in expression with your genes. In the literature for these genes, have you seen or read something that basically shows a big changes in expression, or it's always very small change? Because I'm expecting with these genes that you're not going to see a lot of differences, but I'm not accustomed to these things. So.
There isn't too much literature. It's quite sparse, especially in plants related to the differences or specific studies of these chromomethylase genes, unfortunately. But what we see here in our virtual expression analysis of RT-qPCR, we do see some changes. We don't see it in the short term, but we do begin to see it in the long term. I don't necessarily, I'm not going to come to general conclusions, but I wouldn't expect to see too much of a substantial change in the long term, but I think we would expect to see changes in the methylation profiles and maybe the way these CMT genes are directed in these cold responsive genes in response to the cold stress. So it could be that they're constitutively expressed throughout the gene and they're kind of targeting and going to different areas and changing the methylation profile. So, yeah. Hi. Yeah.
Would you consider well, I know you're looking at short and long-term cold stress, but would you consider doing cold acclimation work, so exposing and then bringing back to normal temperature? Do you think that would influence maybe or be involved in the expression?
I'm just gonna. So is it the question was would we see changes in methylation profile in response to cold acclimation? So exposing the plant to a cold stress and then deacclimating it?
Maybe
yeah.
Maybe not.
So in my long-term experiment that I'm currently doing, I'm going to have a 24-hour deacclimation period in which I return the plants to standard conditions, so 22 degrees. And I'm going to sample and see if there's any changes in expression in that deacclimation period. Yeah.
Cool. And then you bring back down to the cold or
No, there have been previous studies in our lab that have done not specifically CMT genes, but have done recurrent stresses as well as diurnal freezing, in which you expose the plant to lower nighttime temperatures as well as daytime highs, kind of trying to reflect conditions you find in nature in the late fall. And those do see changes in those cold responsive genes, stronger responses in those cold responsive genes and histone profiles in response to that stress. Yeah. Any questions? Any questions online? I don't see any. No? Okay. Yeah.
This is maybe a bit of a follow-up to your own question, but you're not seeing a very significant at the 24-hour point. But is there anything reducing that protein undergo potential translational modifications, which kind of?
That is a great question. And I think that pardon? Oh, the question is, do we expect to see any would there be any post-translational modifications within the plant or the CMT genes, which in response to this cold stress? Initial and over time. That's a great question. I would assume that it could. I would hypothesize that possibly the way in which the plant is directed or the way the genes are directed would change in response to cold stress. But I'm not sure if there would be post-translation modifications. I think a future graduate student, it's like a great future project for a graduate student, but not within the scope of my master's.
The ultimate compound.
Yeah.
I liked your phylogenetic tree, but you didn't include Arabidopsis. Have you tried it with Arabidopsis, and is it the same?
I have in previous 500 trees with single species, and Arabidopsis tends to group within the CMT classes themselves. So Arabidopsis CMT1 would group with CMT1 and it would group with CMT2.
Okay, so
Yeah, it groups it completely. Yeah, but in this case, I kind of wanted to show just monocots.
Sure,
Yeah, except I will note that when I did that, the bootstrap values were quite low.
okay.
Yeah.
So just to be sure, do you think it would be the same for all bicots or like the same pattern? that we would see?
I'm unsure. Possibly, I would assume, but these genes are quite highly conserved, especially those chromo-BAH and C5 methylase regions. So I would assume that they would group accordingly. Yeah. Yeah.
Presentation. So I'm guessing that you basically ordered the mutants at the start of your project.
Yeah.
Right? And so then once you see this information here, especially with this from the JTI, right, would that change the way you approach the project now that you have this information? You didn't have it at the outset.
The question was, would I change my project if I knew that some of the lines did not contain my mutations of interest?
No,
Oh, sorry.
no, this information is right here.
Oh, right here.
About the potential function.
The information regarding potential function and if I had the virtual expression data when I started the project, would I change the direction?
Yeah, thank you.
Definitely. Also, in addition, I didn't include it here, but I've been using AlphaFold to predict the structures of these mutant lines. I didn't include in this project. And I think that would have greatly helped and probably would have refined my selection of CMT genes. Some other directions I would have done in this project is I probably would have looked at the general methylation profiles prior to the methylation profiles of my cold responsive genes. And I would probably change which lines I would have ordered. Possibly, yeah, if that answers your question.
Yeah, because I saw later on you're looking at 3A and 3B.
Yeah.
And based on this graph, I would maybe think, okay, CMT2 would be the obvious choice.
Yeah, I agree with you. Yeah.
Hindsight.
Yeah, hindsight,
Yeah.
but like, yeah, definitely hindsight.
Online? Is there any online?
Any questions online? I don't see any hands raised.
No. All right, well, let's thank Daniel.
Thank you, everyone.
This is the one evaluation sheet. Okay, so is it all clear on that? Okay. And there's not a second percentage delay between the two.
How do I get to oh, there we go.
Yeah, that's 2 out of 15.